Today, most medical therapies aim to treat diseases by targeting abnormal proteins associated with a disease. Usually, these abnormal proteins exist because of mutations in specific genes in a patient's DNA. In the past, we have not been able to alter these genes directly to address the root cause of a disease. However, a novel gene editing system known as CRISPR-Cas9 may allow us to modify, delete, or correct specific areas of our DNA in order to treat these diseases. The system is comprised of two parts: Cas9, an enzyme that cuts DNA, and a guide RNA whose sequence directs Cas9 to a specific location in the DNA where the edit should be made. Cas9 associates with the guide RNA to form a complex that can be easily and precisely targeted to a desired site in the DNA. The CRISPR-Cas9 gene editing process begins when the complex recognizes and binds to a short segment of DNA adjacent to the target site. This initiates unwinding of the DNA helix, which allows the guide RNA to pair with a specific target sequence in the DNA. If the sequences pair precisely, Cas9 cuts the DNA, which disrupts the original DNA sequence and can cause gene inactivation. A larger fragment of DNA can also be removed by using two different guide RNAs to target separate sites on either side of the desired deletion. Cleavage occurs at each site, and the repair process joins the separate ends, thereby deleting the intervening sequence. Corrections to DNA can also be made by adding a DNA template along with a Cas9 guide RNA complex. The template is designed with sequences that exactly match the DNA adjacent to the target cut site. Through a process called homology-directed repair, the cell uses the template to repair the break, thereby replacing the faulty DNA sequence or even inserting a new gene.